Hey, good morning, everyone. This is John Heerdink at Tribe Public. I'm so happy to be here again with all of you, and also to have two new friends in the group here that are pictured with me. I've got Eric Dobmeier, President and CEO of Chinook Therapeutics out of Seattle and Vancouver, and Nipur Lifik, if I could say her name right. I told her I'd mess her name up, but anyway, she is the VP of Investor Relations and Communications at Chinook and is available to you. And I'll get that contact information to you after that in the tribe this week, et cetera, if you have questions directly to her or Eric after this. Again, thank you so much. To remind many of you, Tribe was built over about 4 years ago. We were hosting events across the country in 32 cities. We were bringing companies to the attention of the tribe that the tribe was bringing to our attention through the wish list process. Our belief is that if we come together and collectively, we can better understand how we might move forward in life and make better choices and investments, et cetera. And by getting this direct corporate access to leaders like Eric and brilliant other experts and leaders of NASDAQ and NYC companies, primarily our focus, we're going to become better investors and hopefully better people throughout our life by learning and gaining these insights. Today will be brief, like always, roughly around 30 minutes. I'm going to ask him to tell us a little bit about his background. He's an experienced industry executive in the biotech industry. And from day one, when I met him, after a recommendation by one of you from the tribe that he was building this company and that knew him as a man and as a leader, highly recommended. I've confirmed that from day one, and he's continued to do that in spades. I'm excited about where he's going. Full disclosure, my registered investment advisor, Vista Partners, as many of you know, has invested in Chinook. And that's how we originally took after doing our due diligence, we came to the conclusion that we believed what he was doing and he would be building this very exciting company. And happy to speak to any of you after this at any point and give you my thoughts based on our research. So Eric Dobmeier, will you please tell us a bit about yourself, and then we'll kick off and prompt you to pull up your PowerPoint after that. Thanks so
Great.
much.
Thanks, John. And thanks everybody for being here. Again, Eric Dobmeier, I'm the president and CEO of Chinook. I've been with the company for about 2 years, and I've been in biotech for about 20 years. I started my career at Seattle Genetics in 2002 when it was about a $150 million company. It had recently gone public. And I helped it grow it up to about $10 billion. I was there for 16 years and had a number of different positions, most recently for the last 6 or 7 years as Chief Operating Officer. So that was a great experience. I oversaw a lot of the non-scientific functions, including BD and operations, manufacturing, that type of thing. And then in 2017, I moved on and did a stint as CEO of Silverback Therapeutics, which is another company in the antibody drug conjugate space. So I've spent a lot of time in oncology. And then in 2019, moved on to Chinook. And really, what really grabbed me about Chinook was obviously great science, great investors and people, but also just the huge unmet need in kidney disease that I'll tell you about. It's really an area that's suffered over the past several decades from a lack of drug development, but it's changing rapidly. So that's what really grabbed me, and we'll tell you more about that during the presentation. Thanks. So John, do you want me to just pull it up now and go?
Please pull it up. And just a reminder that Chinook Therapeutics is a public company on the NASDAQ, symbol KDNY is its symbol. Eric's going to get his PowerPoint up here and go through it briefly here. After that, we're going to have a quick Q&A session. If along the way that you want to send me your questions, I'm going to serve them to him as MC to make the process efficient. Just send them through the Zoom chat. Or if you have my email, john@tribepublic.com, please send that either way, and I'll do my best to get the questions answered. Thanks again, Eric. Please begin.
Great. So before I get started, I have to say I will be making some forward-looking statements, so please refer to our SEC filings for more information. Before I get into what Chinook's doing, I want to start by talking a little bit about kidney disease. So it's a huge unmet need. Almost 10% of the world population suffers from some form of kidney disease. And in the US alone, we're spending over $120 billion a year treating these diseases, but most of it is supportive care, dialysis, and transplant. There are very few drugs approved for kidney disease. And there are a number of reasons for this. There are several historical barriers that traditionally have made it difficult to develop drugs for these diseases. Patient populations have tended to be heterogeneous with many different disease drivers. Many of the drugs that have been developed have had non-specific mechanisms, and the trials that have been required have been long, large, and have required hard kidney outcomes. And these are outcomes like progression to end-stage renal disease or doubling of serum creatinine that often take years to progress. But we think a number of things are changing right now in this space. First of all, there's a lot more focus on understanding the underlying biology of these diseases. There's a lot of omics approaches that are coming into the space that have been utilized so successfully elsewhere. And we're coming up with new and better validated drug targets. We've got new translational models to speed up and make drug development more efficient. And probably most importantly, the FDA recognizes that innovation is not where it has needed to be in this space. So they're now open to surrogate endpoints. This is a huge development in the space where we can in some diseases rely on an endpoint like proteinuria, which is a measure of protein in the urine, at 6 months after starting treatment, for example, rather than looking at endpoints that take many years to develop. So proteinuria and eGFR are the two endpoints we're focused on for our two lead drugs in IgE nephropathy. This really speeds up, makes drug development much more efficient, and you can do smaller, more rapid trials. So with all those developments happening, we're really excited about the future for kidney disease. So with the Chinook, we had an extremely busy year last year. In the first quarter, we in-licensed Atrocentin from Abbvie, which is a late-stage program. We're moving into several a phase 3 and a phase 2 trial very soon. In the second quarter, we announced a merger with Atara Biotech, which brought in Bion-1301. Atara was an immuno-oncology company, but had one very interesting kidney disease asset, which grabbed our attention. So through the merger, we brought in a program that's in phase 1B right now into our pipeline. Then in the third quarter, we raised $115 million from some of the most prominent healthcare investors in the space. And in the fourth quarter, we closed both the merger and the financing and became a public company, as John said, traded on NASDAQ under the ticker symbol KDNY. So now we have all the building blocks to achieve our goal of building a leading kidney disease company. I see an opportunity here through this pipeline to do something similar to what we did at Seattle Genetics, take these really exciting programs, advance them in a very smart, efficient way, and develop a strong pipeline of programs for diseases that really have very few treatment options. So Atrocentin here is our lead product. It's in late stage development. We believe it's the most potent specific ETA antagonist. We believe it's best in class. It'll be moving into several trials that I'll tell you about very shortly. Bion-1301 is an anti-APRIL antibody. We've got quite a bit of healthy volunteer data that's been reported that looks very good, and we're now in patient treatment in patient cohorts for this program in phase 1B trial, and we'll have multiple data sets to report out at various nephrology conferences during this year. And then CHEK 336 is our first internally developed program. This is an oral small molecule for a disease called hyperoxaluria. This is a kidney stone formation disease with very few treatment options. We think with a well-tolerated oral small molecule, we can treat many different types of this disease that I'll tell you about. And we have a phase 1 trial plan to start before the end of this year. We're in IND enabling studies right now, which are going well. And then underpinning all of these programs and the future of our pipeline is a precision medicine approach. So this is an approach that's been used very effectively in oncology and rare disease and other areas. So we focus on rare severe chronic kidney diseases. We're designing novel, targeted, and differentiated molecules that go after key drivers and pathways in these diseases. And then we're doing clinical trials in defined patient populations with surrogate endpoints that speed up and make drug development more efficient. And we also have a very strong cash position with operating capital through through the first half of 2023. So we're in a great position to advance our pipeline. So this is a snapshot of our pipeline. Atrocentin is focused on a phase 3 in IgA nephropathy called ALIGN that will start very soon, as well as a phase 2 basket trial, which is a life cycle management and signal seeking trial that'll start soon after ALIGN in a number of different glomerular diseases. We've also got BION-1301 in a phase 1b in IgA nephropathy patients advancing well, and then CHEK 336 moving towards the clinic, as well as research and discovery programs in polycystic kidney disease and other severe chronic kidney diseases. We own commercial rights to all of our pipeline programs globally. We plan to commercialize our drugs in the US and probably Europe ourselves, and then may look for partnering opportunities in Asia, where a number of these diseases are very common. We don't have aspirations to commercialize ourselves in Asia, but think there could be some really interesting partnerships there. And we're also looking for additional opportunities to continue building our pipeline through both precision medicine platform add-ons as well as preclinical programs. So before I get into our products themselves, I want to talk a little bit about IgA nephropathy as a disease. So this is an autoimmune condition. It's the most common primary glomerular disease, and by that, it affects the glomerulus of the kidney. And there's about 140,000 patients in the US, several hundred thousand in Europe, and several million in Asia where it's more common. It is an orphan disease in the US and Europe, which provides exclusivity advantages, but it is not that rare overall. There are no approved treatments for this disease, nothing. The way it's treated currently is with blood pressure lowering medications and immunosuppression, approaches like steroids and even chemotherapy drugs, none of which work particularly well. The most important predictor of progression in IgA nephropathy is proteinuria. That's the amount of protein being deposited in the urine. And when the kidney is damaged, its filtration properties are less effective, and you get higher levels of protein in the urine. It's a marker of progression for IgA nephropathy. And over a gram per day of protein is a strong predictor of progression. The FDA has recognized this and is now supportive of proteinuria as an accelerated approval endpoint with eGFR, which is estimated glomerular filtration rate. It's a measure of kidney function as the full approval endpoint. And when you look at how the disease progresses, it's not well understood why patients develop this disease, but it often follows a gastrointestinal or respiratory infection where a patient starts making higher levels of galactose deficient IgA. These are aberrant IgA molecules. And then the patient mounts an immune response to those molecules, and they form immune complexes. So they're these immune complexes that circulate and are deposited into the kidney where they cause damage by increasing pressure and causing inflammation and fibrosis. So with our two programs, Atrocentin and BION-1301, we have two very distinct mechanisms of action for IgA nephropathy. BION-1301 is potentially disease modifying. It can prevent the formation of these immune complexes in the first place so that they're never deposited in the kidney and don't cause damage. Whereas Atrocentin can reduce the damage caused by the immune complexes once they're deposited through its vascular, anti-inflammatory, and anti-fibrotic effects. So initially, these drugs could be used in different segments of the patient population, but we also think they could be very effectively combined over time. And we think the future of IgA nephropathy is similar diseases like follicular lymphoma or other diseases that used to be fatal in many cases, but now can be managed and become chronic with multiple lines of therapy and multiple combination regimens. So let me tell you a bit about Atrocentin. This is a drug that Abbvie developed for diabetic kidney disease in over 5,000 patients. It's got picomolar potency, and it's highly selective for ETA over ETB, which is important in terms of minimizing fluid retention, which is really the only significant side effect of this drug that's been observed. The optimal dose was established through multiple phase 2 trials at Abbvie. And you can see here on this slide in the chart, this is from a trial called RADAR, which was a phase 2 trial that Abbvie ran in diabetic kidney disease. And you can see that when patients go on Atrocentin, their proteinuria or albuminuria drops very quickly within the first week or two and stays down well on drug. And what they observed in SONAR, which was an over 5,000 patient trial, is a similar 30 to 35% reduction in proteinuria, which is a very significant reduction and would be expected to translate into long-term kidney protection. They also saw hard kidney outcome improvements in the SONAR trial. There's a long history of what happened with SONAR, but the short story is they started the trial in 2002. They were treating patients for 5 years. In the meantime, they had a strategic change of direction at Abbvie, and they also got impatient about the pace at which the events were happening in the trial. Along the way, they ended up giving bardoxolone back to Reata, and they closed down the SONAR trial and didn't know what the data was at the time. 2 years later, when the data was analyzed and reported, both at the World Congress of Nephrology and published in Lancet, it was a positive trial. So in 2020 hindsight, if this trial had been continued, it likely would have led to an approval. But it was really a change of a strategic direction at Abbvie. We were able to come in and convince Abbvie that the ideal indication for this drug is IgE nephropathy, where patients are young and healthy and really have single organ involvement and are otherwise don't have a lot of comorbidities, compared to diabetes, where there are a lot of things going on. So the safety profile of this drug is well established in over 5,000 patients treated for up to 5 years. And based on the proteinuria reductions and the eGFR benefits seen in that trial, we think we have a really strong rationale for taking this drug into IgE nephropathy and other glomerular diseases. So what we're planning to do next is start the ALIGN trial very soon. It's a phase 3 trial for patients with biopsy-proven IgAN. They'll all be on frontline standard of care, which is a blood pressure lowering medication, an ACE or an ARB that's been optimized and on a stable dose of that. They'll all have proteinuria over 1 gram per day and maintain kidney function of at least 30 mils per minute in terms of eGFR. It'll be about a 300 patient trial, 1-to-1 randomization. It'll be run globally at 140 sites roughly in 15 to 20 countries. And the primary endpoint is proteinuria at 6 months after starting treatment, with the full approval endpoint being eGFR, kidney function, measured at 2.5 years. So the trial starts soon, and then we've guided to having data, top-line data on proteinuria in 2023. We'll get more granular with that guidance once we have more information on enrollment rates, because that's the main driver of the timeline. In parallel with the phase 3, we're starting a phase 2 basket trial called Affinity, which will start soon after ALIGN. This is an open label study with 4 initial cohorts of 20 patients each. The primary endpoint is proteinuria at 12 weeks. And we're going to overlap this trial with some of the sites for the phase 3, particularly for this cohort of patients with lower proteinuria. So these are IgAN patients with between 0.5 gram and 1 gram per day. So slightly less proteinuria, but still at risk of progression. That's one cohort we're doing. We're also going to test it in a kidney disease called FSGS, as well as Alport syndrome. And then we're going to do a combination of Atrocentin with SGLT2 inhibitors. This is a class of drugs that are used in diabetes and also starting to expand into other chronic kidney disease. We have some very interesting combination data from the SONAR trial, where 14 patients got Atrocentin and an SGLT2 inhibitor, and the data looks fantastic. More proteinuria reduction and less fluid retention than Atrocentin alone. So we're starting Affinity soon after ALIGN in the first half of this year. We'll be able to start reporting data in 2022 because it's an open label trial. We'll have multiple opportunities to present data during 22 and 23 while ALIGN the data is maturing on ALIGN. Moving on to BION 1301. This is an antibody that targets the APRIL. We think this is an ideal target in IgA nephropathy. APRIL is a TNF family cytokine. It's involved in B cell signaling and drives both production of both normal IgA as well as galactose deficient IgA. And it also drives survival of IgA secreting plasma cells. So by depleting APRIL, we believe it's a strong way to deplete both normal and galactose deficient IgA levels to reduce the formation of these immune complexes that drive the disease. Higher APRIL levels are correlated with worse prognosis, higher proteinuria, lower eGFR in this disease, and APRIL gene variants also confer increased risk of IgAN. So it's really an ideal target for this disease. Our antibody has been tested extensively preclinically in both non-human primates, in both IV and sub-Q. And we've also done a number of safety studies in healthy volunteers, including dosing up to 2,700 milligrams in a multiple myeloma study that was done by Aduro in the past. So what we've done so far, what we've reported from this program is data from the single ascending dose and multiple ascending dose cohorts in phase 1. And what you see here on this slide is BION-1301 was really well tolerated with no severe adverse events. The half-life was over a month, so it was a very well-behaved molecule. We saw dose-dependent and durable reductions in free APRIL levels, as well as IgA and IgM levels, but less effect on IgG levels, which is important in terms of minimizing infectious risk. In this disease, we haven't seen infections as a side effect in healthy volunteers, which is promising. We also importantly saw 50 to 60% reductions in IgA with doses of 150 to 450 milligrams. And this has been shown with another molecule called atacicept, this level of IgA reduction to translate into significant proteinuria reductions. So we're really enthusiastic about what we're seeing so far. We'll have a bunch more data in the first half and second half of this year. So in the first half, we'll have data from a study we're doing on bioavailability of 1301 to look at IV to sub-Q formulation. So we're doing IV right now. We're gonna plan on using sub-Q for our later stage trials. So we'll have some biomarker data from that trial to report. We'll also be able to report the galactose deficient IgA levels in the healthy volunteers, which have not been reported yet. And it's a very close biomarker to predict impact on proteinuria. And then we'll have the early data from the patients in part 3 of this phase 1b study. This is IgA nephropathy patients treated with BION-1301. So we'll be able to show biomarker data as well as early proteinuria data from those patients. So a lot coming from BION-1301, and we're really excited about the mechanism of action here and the ability to really be truly disease modifying in this disease. And then our third program is CHEK336. This is a small molecule that we're developing for hyperoxaluria. So these are diseases caused by excess oxalate. Primary hyperoxalurias are genetically driven. PH1 is a very severe form of this. It's an ultra rare disease. And through a liver enzyme deficiency, it causes overproduction of oxalate. And this forms kidney stones, multiple kidney stones, that can even lead to kidney failure in the most severe cases. With PH1, which is the most severe, the median age of kidney failure is 23 years old. And the only treatment traditionally for this disease has been combined liver and kidney transplant. If you just transplant the kidney, the stones come back. So you have to transplant both organs, which is obviously a significant intervention and very expensive and life-altering for these patients. There's about 5,000 to 7,000 PH1 patients in the US and Europe. PH2 and 3 are less well understood and diagnosed, but thought to be, there could be quite a few more patients than that in those diseases. And then even more so, secondary hyperoxalurias are much more common. And these are diseases that generally occur after gastric bypass surgery or other GI abnormalities. They can also be idiopathic. Sometimes we don't know what causes people to form frequent stones. And we think that there's opportunity through a well-tolerated oral small molecule to have an impact across the board in hyperoxaluria. So with CHEK336, we've done a number of preclinical studies here. You can see this chart here on the left shows a PH1 mouse model where we saw significant and dose-dependent reductions in urinary oxalate with our molecule. We saw wide safety margins in tox studies. And we believe that the titration and customized dosing that we can do with this drug may be a really attractive property. There are some other drugs from Alnylam and Dicerna that are siRNA molecules. They're injectables, but they can't be titrated and customized for dosing. And a number of patients stay in the above the normal range of oxalate with those approaches. Whereas with oral administration, which is more convenient, we may be able to really dose per patient to get the oxalate levels down in the normal range and reduce the number of stone events. We can also potentially move into less severe, more common forms of hyperoxaluria. So this drug is in IND-enabling studies, which are going well. It should be moving into the clinic before the end of the year. Financially, we're in great shape. We had about $290 million in cash and investments when we closed the merger with Aduro in early October. This cash should last us through the first half of 2023. And currently we have a little over 42 million shares of stock outstanding. From a catalyst point of view, number of things going on this year. We'll be starting the phase 3 ALIGN trial very soon, and soon after that, the phase 2 AFFINITY trial. we'll be presenting multiple data sets that I described for Bion 1301 in both the first and second half of the year, and then making some decisions on the next stage of development for that program. And then with Check 336, we'll be moving into the clinic by the end of the year. So we think we've got all the ingredients to build a leading company in this space. We're laser focused on kidney disease and executing on this pipeline. We've been on a really rapid pathway last year, and we don't intend to slow down anytime soon. So we think that the future is really exciting for Chinook. So with that, I'll turn it back over to John for some Q&A.
Sure. Maybe we'll just reduce the PowerPoint for a second here. Thank you, Eric. Very interesting. Again, I found this through one of the tribe members, if you will, back in the summer when this was all getting put together. And what struck me was after taking a fairly deep dive with some people that are way smarter than me in the scientific community that I have the pleasure of knowing, everybody came back in spades and said, this is very, very interesting. I like the late stage assets. I like the breadth, and I like the focus. It's really and truly, relatively unmet need here. And the other thing that came across is the management. They came across in spades. 100% came back to me and said they like Eric and Eric's approach, and he surrounded himself with some very solid people and very capable. One of the questions that one of the tribe members had was, what is the current insider ownership after putting this company together? Where do you stand, Eric? Where do the insiders stand?
Yeah, so our Chinook was funded by three venture capital groups, so Versant Ventures, Apple Tree Partners, and Samsara. And we still have board members from both Gerald Davis from Versant and Srini Akkaraju from Samsara are on the board. So and they own a significant amount of the company. Versant's our largest shareholder, I think around 20%. And then I think Samsara is close to 10% ownership. The management team has significant ownership as well. I think my ownership's in the 3% range, and then we have a number of other executives. And really, I've been buying in the open market, as you can tell from the filings we've been making. I think we're really undervalued right now, and I have no intention of. I don't think anyone on the management team feels like now is the time to sell. We think there's a lot of upside here from where we are, and I think. In kidney disease in general, there's a number of developments happening. There's several drugs that are moving towards either data readouts or approvals. I do think that the adage "a rising tide lifts all boats" applies to this space because there's kind of a discount now on kidney companies versus oncology or rare disease, which are much well understood and have had much more attention. So I think with increasing progress in the space, it's going to help all kidney disease companies, and there's a real strong belief among our insiders that the future is bright and there's a lot of upside from here.
Very good. A follow on to that is that the 115 million that came in on the consummation of and moved to NASDAQ. You also reverse split the stock from the sort of 270 range at the time, if I remember correctly, and 5 for 1. And yep. That put us sort of in that 11-12 range, and the stock has legged up north of 20 a couple times as it started trading as kidney, KDNY officially. And then you've seen the sell side support come into the stock that I've seen and roll out coverage. I believe there's 7 analysts that are covering you, have a price target between 25 and 38, depending on who the analyst is. Can you speak to that support that you have there and what analysts are covering, or is that a Nupur question, or who can address that for us?
Yeah, maybe I'll start and then hand over to Nupur. I've been amazed at the support from the sell side. I mean, we've got 7 analysts to initiate in less than 3 months is pretty amazing, and it's a testament to the story, but also the work Nupur's done. So it's been fantastic. Nupur, do you want to comment with your thoughts on our analyst coverage?
Yeah, absolutely. The coverage has been overwhelming, and it's been great. I think the team has done a great job of sort of educating the analysts even before I joined. So I headed up investor relations for Enduro and joined the team in October around the time that the merger closed. So there's just been conversations going on for quite some time, and all of our analysts are very enthusiastic. So we have coverage from Cantor Fitzgerald, Emma Nealon, Evercore, we have Josh Schimmer, H.C. Wainwright, Ed Arce, Oppenheimer, Justin Kim, SVB Leerink, Joe Schwartz, Wedbush, Laura Chico, and William Blair, Matt Phipps. And so, some of these analysts have experience covering multiple kidney disease companies and others are new to it, but they all share that enthusiasm for what we're doing. So it's been exciting.
Thank you. And then just a reminder to the tribe, you can send questions to me for Eric through the Zoom chat feature is the quickest way to do it during this event. The one question that just popped up, it says, you spoke to, regarding Abbvie, is there an ongoing relationship of any kind with Abbvie and in the commercial scope, co-development, or is there strictly a license arrangement? Can you speak to that arrangement?
Yeah, Abbvie it's a standard license agreement. So basically, we have full rights to the program, but we will, the deal was structured to have small cash upfront. Abbvie got 15% of our equity at the time, which has now been diluted down to less than 5%. And then we owe 135 million in milestones that don't start until after approval. So they're all commercial and approval related milestones. And then royalties that start in the high single digits and go to the high teens. But Abbvie does not have any commercial rights or co-development rights. It's full ownership and control by us with the subject to payment of the milestones and royalties to Abbvie. They've been really supportive. There is a strong relationship in terms of supporting tech transfer because they spent many years developing this drug. We got quite a bit of drug in vial to use for our trials from Abbvie. So we do have a relationship with them, but not in terms of co-development or future rights to the program.
Okay, thank you. And then I guess one of the questions is, within the kidney space, how are you differentiated in your approach composed to anyone? How would you address that in general?
Well, in the kidney space, there are some big pharmas that have played in this space, and they tend to come in and out depending on how excited they are about it, and mostly focused on diabetic kidney disease because diabetes causes about 50% of kidney problems that we experience in our society. And so they focused on the large indications. And then there are some smaller companies, but they tend to be more rare disease companies that have one product. I'm thinking about like Reata or Travere or others that are really more focused in the rare disease space and maybe have one kidney program. I think what differentiates us is the focus on kidney disease and the breadth of our pipeline across different kidney diseases and the precision medicine approach and commitment to continuing to play in this space. So we're really trying to develop deep long-term relationships with the nephrology community by having multiple products. We've got 2 products in IgE Nephropathy, which is more than anybody in the world, I think, in terms of a focus on that disease. So we're truly a kidney-focused company. I think the only other company I would really say that's doing what we're doing is Goldfinch, and they're a private company currently and a little bit earlier in development. I think they're just moving into phase 2. But that's, I think, what differentiates us, as well as, the strong institutional support, the great science that we're doing. There's a lot of things that are real positives about this company that I've been impressed with since I joined.
Great. Speaking of institutional coverage, I believe the fund, the round that came in, 115 million, was led by EcoR1. And could you speak to that, the institutions that were involved there that is in the public domain?
Yeah, so EcoR1 and OrbiMed were the 2 sort of largest participants in that financing. We did press release the funds that would let us use their names, and Rock Springs and Fidelity were included. Newport, I don't know if you have the whole list in front of you, is probably 10 or so investors that participated in that financing.
Yeah,
but there were a number of really big names that were not disclosed, and the 13 Fs I think come out in mid-February, so there'll be more information out there on who participated in that financing within a few weeks. But it's a who's who of biotech investors.
And so, what is left in the float ex insiders share count? Do you have?
Do you know that answer, Newport? I haven't looked at the specific number.
The public float, no, not off the top of my head. We do have, I think, 42.2 million shares outstanding in terms of common stock, fully diluted around 46 million. But public float, we can get back to you, John, to let the tribe know.
Well, it sounds like with Versant, we can take out 20% of that flow because they're a 20% holder. So that's an easy.
Yeah, I think if you add up Versant, Appletree, Samsara, and Morningside Ventures was the largest shareholder in Aduro, plus the insiders, you're probably getting to about 50% ownership from that group. And then the other 50% is, a lot of large institutions currently hold the stock.
Yeah, it sounds really a very tight float at the end of the day, because that's down to 20 million or so, and then you chunk out bigger institutions that came in that 115 million that may not be insiders, may not be 10% holders, but they're known and generally for putting in significant capital, looking for 2 to 3 horizons. And as you mentioned, with 290 million or so, you are funded through sometime in 23, right? So that kind of goes along with what I understand is what sometimes I call them as the biotech mafia, if you will. Not, maybe they don't like that, but they're brilliant investors in the space, noted. They do these club deals and they find projects and companies like yourself and get yourself funded through significant milestones. Speaking of the milestones, again this year, can you just this year line that out for us succinctly?
Yeah, so the phase 3 and phase 2 trials of Atrocentin will start soon. So those are important milestones for us to get the trial started. We'll have a number of data sets to report out from Bion 1301, both healthy volunteer and patient data. We'll also have some really interesting preclinical data on Atrocentin, identifying and defining these anti-fibrotic and anti-inflammatory mechanisms that are less well understood. And then we'll be moving 336 into the clinic. There's also some BD activities that are ongoing to bolster the precision medicine approach that we're taking, as well as interest from Asian companies in geographic territorial deals for Atrocentin and even 1301. So we don't have aspirations to commercialize in Asia ourselves, so we'll likely do a partnership there. And then moving into 22, that's when we'll start having serious data flow from 1301, from Atrocentin in the phase 2 trial, as well as probably some data from 336, and then moving into the phase 3 results in 2023. So we feel like we're really on the cusp of some major driving catalysts for the company.
Yeah, it does sound like that. One last question here before we sign off. As far as building this company, you have a breadth of programs, no doubt. Are you looking to be acquisitive or develop, what does your R&D situation look like? How would you speak to that?
Yeah, I think, we've obviously brought in both Atrocentin and Bion 1301 over the last year. So, if we saw an opportunity like that again, we would absolutely go after it. There isn't as much competition in kidney for assets, but there are fewer assets than like oncology or other areas. So we'll see. I think we're going to be more focused this year on precision medicine technologies, as well as preclinical programs and targets. We do have several research and discovery programs ongoing in both PKD and other diseases, but it's always helpful to supplement that with external BD. But I doubt we're going to be doing the kind of BD that we did last year again.
Yeah. No, you certainly have a lot of action over the last year. So very interesting presentation today. I thank you, Eric and Atrocentin, for joining us. I thank all of the tribe that have joined us, and we'll look to do in the future. Note, tribe, that again, this is the process of the wish list. If you want to see Eric again present, please submit your wish list interest every week, and all the other companies as we send out the tribe this week on Fridays at 1:15 Pacific, 4 p.m. Eastern, the weekly newsletter where we connect and keep you up to date on the companies you care about. And if you have any other questions that just didn't come to mind through this, please send those to me at john@tribepublic.com. I'll also get you the contact information for Deborah on the call today so that you can reach out to her if you like. The lastly reminder, if you want to review this again, we're going to have the video up by the end of the week at the Tribe Public YouTube channel. Just do a search if you haven't been there before at Tribe Public. At Tribe Public, and you'll find it, and the other videos of the events that we've hosted here, of these brilliant leaders. And I thank you all for your attendance. Thank you, Eric. Thank you, Nipur, and thank you, Chinook. And we look forward to seeing you again soon. Thank you so much.
Thanks a lot.
John.